You know, from the yogurt in your fridge to the medicines that keep us healthy, so many products in our lives come from this hidden world of microscopic manufacturing. So today we're going to pull back the curtain and see exactly how it's done. We're going to follow one single molecule on its journey, all the way from a single cell to a final purified product. And our journey starts with a simple question.
So take a look at these. We've got yogurt, right? Something you eat for breakfast. Then you've got alternative fuels, stuff that could power our cars one day. And then life-saving drugs. I mean, they seem totally unrelated, don't they? But they all share this one kind of remarkable common origin. And here it is. They are all made in microscopic factories. Yeah, we're talking about living cells, things like bacteria, yeast, even mammalian cells, that we've basically hired to manufacture specific products for us on a massive scale. The fancy name for this is fermentation. It's a process we've used for centuries to make bread and beer, but now it has been totally supercharged to become the engine of modern biotech. Okay, so our journey begins with the first big stage, which in the biz is called the upstream process. Honestly, the easiest way to think about it is like farming. But instead of growing corn in a field, we're growing trillions of these tiny cellular factories in a super-controlled environment.
So what is fermentation really? Well, it's a process where we take a specific type of cell, we feed it this nutrient-rich diet, and we just let it grow and multiply to produce something we want. And for our journey today, we're going to follow the production of a really cool molecule called green fluorescent protein, or GFP for short, which is made inside E. coli bacteria.
Now, you don't just start with a giant vat of cells. No way. It's this really careful scaling up process. It all starts with a teeny tiny seed stock of our E. coli, thawed from a freezer and put into a small flask with some liquid food, or media as they call it. Once that population grows, you transfer it to a bigger flask, and then an even bigger one. And you just keep repeating that until we have enough of these microfactories to start the main event in a massive production bioreactor.
Inside that bioreactor, the cells go on this pretty predictable roller coaster ride. First, there's the lag phase, while they're just getting used to their new home. Then boom, the log phase. This is the population explosion we want, where they're doubling at an incredible speed. But, no party lasts forever. As food starts running low, they hit the stationary phase, where growth just stalls out. And that is our sweet spot. We harvest right there, at peak productivity, before the inevitable death phase kicks in and we start losing our product.
But listen, none of this just happens by accident. That bioreactor isn't just a big tank. It's a perfectly controlled world. Every single one of these variables, temperature, pH, pressure, oxygen, is monitored constantly, in real time. It's precision engineering on a biological level, all designed to keep our microscopic factories happy, healthy, and making our product.
All right, so our cells have done their job. They've manufactured our green fluorescent protein. Awesome. But there's a catch. The product is locked inside them. And that brings us to stage 2, recovery. It's time to crack these cells open and get the prize. And to do that, you gotta understand this key difference. Some products are extracellular, which means the cells kind of spit them out into the surrounding liquid. If that were the case, you just keep the liquid and toss the cells. Easy. But our GFP is intracellular. It's kept inside the cell. And that means we need to collect the cells themselves and then figure out how to break them open.
And this is how we do it. The whole fermented broth gets spun in a massive centrifuge to separate the solid cells from all that used-up liquid. Then, the cells we've collected get a good wash to get rid of any gunk. The next step, homogenization, is the big one. This is where we physically bust the cells open. Afterwards, we centrifuge it again, but this time we throw away the solid cell junk and keep the liquid, which now has our GFP in it. A final filtration step just cleans it up a little bit more. It's basically a whole series of mechanical steps. Separate, wash, break, separate again, all to get us that precious liquid.
And to really get a picture of what's happening during homogenization, the source material just puts it perfectly. The cells are pumped at an absolutely insane pressure. We're talking 13,000 pounds per square inch through a tiny, tiny opening. And just like a balloon being tightly squeezed, the cells can't take the stress and they rupture and break apart. It is a powerful mechanical way to get that prize out.
So after all that separating, washing, and breaking, what do we actually have? We have a liquid called clarified lysate. But here's the thing, it's not pure GFP, not even close. It's more like a biological soup. It's got our target protein in there, but it also has a whole mess of other stuff from inside the cell. Other proteins, DNA, you name it.
And this takes us to our final and maybe the most amazing stage of all,
purification. The challenge now is to go into that complex, messy biological soup and pull out one specific type of molecule, and only that molecule, leaving everything else behind.
The main tool for this job is called chromatography. So I want you to imagine a big column packed with these tiny microscopic beads. Think of it like a super specific molecular filter. These beads are designed to be sticky, but only to one type of molecule. As our biological soup flows through, our GFP gets stuck on the beads, while all the other junk, the other proteins and debris, just washes right on by.
So the purification process is kind of like a gauntlet with multiple checkpoints. First, the liquid goes through a column that separates molecules by their electrical charge. Our GFP has a specific charge, so it sticks while others don't. Then we change the liquid a bit and send it through a second column, this time one that separates molecules based on whether they hate water, kind of like oil. Each step is like a finer and finer sieve, just removing a different set of impurities and getting us closer and closer to a pure product.
And that last step uses this really clever tech called tangential flow filtration, or TFF. Okay, think about a normal filter, like for your coffee. You push water through it, and eventually it just clogs up, right? Well, with TFF, the liquid flows across the surface of the filter. This cross-flow acts like a little squeegee, constantly cleaning the surface and preventing clogs. It's way more efficient for these big industrial processes.
So TFF is a real workhorse here, doing two critical jobs at the same time. First, it removes water to make our product way more concentrated. At the same time, in a process called diafiltration, it's also washing away the old liquid and replacing it with the final stable storage buffer. It concentrates and swaps the buffer all in one go. Pretty smart. So there you have it. We've actually followed our green fluorescent protein all the way from a single cell to a pure final product. And when you really boil it all down, the entire incredible journey is made up of three core stages. First, fermentation, where we grow the cells that become our little factories. Then recovery, that whole mechanical process of harvesting the cells and cracking them open. And finally, purification, the super precise chemical process of grabbing our one target molecule out of all that other mess.
So what's the big takeaway here? Well, it's the sheer power of fermentation to scale up from almost nothing. It's the brute force necessity of recovery to get the product out. And it's the chemical magic of purification to get something so unbelievably pure. But maybe the most important thing, the thing that underpins all of this, is that none of this happens by accident. Especially when you're making medicines, where purity is literally life and death. Every single action, every temperature check, every measurement is all guided and documented in something called a batch process record. It's the master recipe that guarantees every single batch is safe, pure, and consistent.
This whole process has given us everything from new medicines to better biofuels. It's really a testament to our ability to partner with biology on this huge industrial scale. And it leaves us with a really fascinating question to chew on. Now that we have the tools to build molecules with this kind of precision, what are we going to choose to build next?